we're going to begin looking at the cell in general. There's something called the cell doctrine. The cell doctrine states that all living things are composed of cells and cell products. So even if you are a single-celled organism, you would of course be considered to be alive. A single cell is the smallest unit that exhibits all the characteristics of life. So like I had mentioned, a single cell would in fact be considered to be alive. And all cells only come from pre-existing cells. So what we're saying here is there's not spontaneous generation of a cell. A cell has to be cloned and or reproduced to create another cell. There are a couple different types of cells. The first one that we're going to look at is a eukaryotic cell. Eu means true, karyo refers to the nucleus. So eukaryotic cells are going to be those that have a true membrane-bound nucleus. So by definition, the eukaryotic cell, of course, contains that membrane-bound nucleus. All eukaryotic cells are going to have a couple different structures within them. One, of course, is the nucleus, where we have the DNA. Another is the plasma membrane. That's going to be the outer boundary to the cell. And the third is the cytoplasm. That's going to be the semi-solid material inside of the plasma membrane, and that's where the majority of the organelles are going to be found. The cytosol then is mostly the liquid component of the cytoplasm. And as I had mentioned, the other thing that you'll find floating around in the cytoplasm are the organelles. This is a generic eukaryotic cell. As you look at it, the center structure that's in purple is going to be the nucleus. The outer membrane that's in yellow would be the plasma membrane. And then you can see various different organelles inside the cytoplasm. Prokaryotic cells, on the other hand, pro is going to be before, karyote again is going to be nucleus. So prokaryotic cells literally mean before nucleus. They have a plasma membrane, and that plasma membrane is surrounded by a rigid cell wall. Bacteria are really good examples of prokaryotic cells. So if you take a look here, we've got a eukaryotic cell on the left and prokaryotic cells on the right. The eukaryotic cell, you can see the large circular structure in the center, that's the nucleus. The outer membrane, of course, is the plasma membrane, and then again, various organelles inside of the cytoplasm. On the right-hand side, you don't really see any membrane-bound nucleus in the prokaryotic cell like you did in the eukaryotic cell. In addition, you can really see that rigid cell wall fluorescing in blue around the outside of the prokaryotic cell. That structure is lacking in the eukaryotic cell. Cells need to maintain a high surface area to volume ratio. That is going to allow them to undergo metabolic processes efficiently. In other words, in order to get raw material into the cell, we need to have lots of surface area and low volume. Think about that for a second. The further you have to travel through the cell to get, say, to the nucleus, to get materials to the nucleus, the less efficient that cell really is. On the flip side of that, the further distance you need to travel to get rid of wastes is also going to play a role here. So we want to have a small distance really to get materials in or out of that cell, and we do that by maintaining a high surface area to low volume ratio. The way we do this is by division. So if our cell gets too big and the distance is too far to get materials in or out, we're simply going to have the cell divide, and then the resultant cells will be smaller and maybe more efficient. So as we were saying, if you look at cell A, one large cell, the distance to get materials in, say, to the center of that cell is significantly high. But if we take that one cell and we break it down into 8 smaller cells, the distance now to get to the center of each of those resultant cells is much smaller. As a result, the cell is more metabolically efficient. The other structure found on some of the cell surfaces is something called microvilli. Microvilli are present in order to increase surface area as well. They're very, very small finger-like projections that I liken really to a terry cloth towel, how it has all the loops inside of the towel for absorption.